quiz 7. It's available to you all day. Know that some people have taken it already, and those of you who haven't, don't forget. Second thing I wanted to mention is that I posted in Canvas a worksheet on enzymes and catalysis, which is the next topic that we're moving into. And that worksheet is due next Tuesday so that we can discuss it in class next Wednesday, a week from today. So you should be able to see that this will be a graded worksheet, so you'll see it as an assignment in Canvas. And finally, I looked up when the final exam will be, and it turns out that we could have it either early. The final exam week is June 1 through 7. So we could have our final exam either on Wednesday, June 2, or Monday, June 7. In other words, it could either be early in finals week or on the last day of finals. So I would like your opinion if you have one. If you don't have one, I'll just choose one of those. If you do, send me an email. And if there is a vote, I will go with whichever one of those options gets the highest number of votes. So again, it's either Wednesday, June 2, or Monday, June 7. And again, you will have all day. On whatever day we choose, you'll have all day to take the exam. It will be 2 hours in length. Okay, any question about any of the things I've said so far?
Professor, how many questions will there be?
Don't know. Haven't given any thought yet to it. Okay. But what do we usually have 20 or 25 for 1 hour, so I would guess twice that many.
Okay, thank you.
And it will cover the entire semester, of course. All right, I'm going to turn to the topic of the day. And first, so we're going to talk today about enzymes and catalysis, but I want to give you an overview of what's happening in the bulk of the remainder of the course. We're going to be talking about the 4 topics that you see listed here: energy, enzymes, photosynthesis, and respiration. And they're all interconnected. And this will become clear as we go along. The first we'll consider energy. What is it? Where does it come from? How do life forms use it? Enzymes, we'll talk about enzyme structure, enzyme function, and the mechanisms by which they act, and very importantly, how they're regulated. We don't want an enzyme to be active all the time. So how does the cell turn it on and turn it off? Then we're going to talk about photosynthesis, which is the mechanism by which the sun's energy is captured and converted into chemical energy. And that's how we get food. By conversion of the chemicals that are generated in photosynthesis, we can extract energy from those chemicals, and that is what we live on. That's our food. Respiration is, in a sense, the opposite of photosynthesis. It's how we derive energy from food. So this is to give you the 30,000 foot view of the rest of the course. And now we're going to talk about specifics. Energy and enzymes. So the one thing we can count on is the sun. The sun is a star, shines all the time, constantly bombarding the planet Earth with energy in the form of light. We have organisms on Earth that can capture that light. One of them is not a butterfly, but rather the plant that the butterfly is sitting on. The plants, most everything on Earth that is green, is a producer because it can capture sunlight, and everything else is a consumer. It either eats plants or eats plant products or eats organisms that have eaten plants. So what is energy? It's the capacity to do work. What is work? Work is the ability to move matter in a direction that it would not move if it were left alone. Energy is measured in calories. The definition of a calorie is given here. It's the amount of energy that it takes to raise the temperature of 1 gram of water, 1 degree centigrade at sea level. And the elevation is important in determining the amount of energy that it takes. Now, you're all familiar with calories, but probably you're mostly familiar with calories as they relate to diet. A dietary energy is not measured in calories, but rather in kilocalories. 1 kilocalorie is 1,000 calories. So if you're looking at a 2,000 calorie a day diet, that's really 2 million calories per day. Forms of energy, potential and kinetic. And this becomes very important when we talk about enzyme action. So potential energy is stored like a rock sitting at the top of a hill, or charged ions on one side of a membrane. And in that regard, we will talk about an event called chemiosmosis, which is where we get a very large part of the energy we use comes from the process of chemiosmosis. And that has to do with ions passing from one side of a membrane to the other.
I have a question really quick.
Yeah.
Is there a difference between calories with a capital C and calories with a lowercase c?
I don't know. Scott, do you know?
If I can, I know. Big C calories is short for kilocalorie. That is the calorie that we know, whereas little c calorie is the, in the case that was given, the 1,000 calories, that would be the million calories, the small c calories.
Okay, thank you.
So small c is the generic term for calories. Large C is dietary calories.
Effectively, yeah.
Okay. All right, thank you. All right, back to kinetic energy. That's energy in motion. So you're imagining the rock sitting on a hill. When it tumbles down the hill, it's exerting kinetic energy, or water driving a water wheel. And again, chemiosmosis, when ions move through a membrane, they charge an ADP, excuse me, an ADP molecule to an ATP molecule. And that's where we get the bulk of the energy for metabolism. So potential energy and kinetic energy are being constantly interconverted in biological systems. Now, thinking about these things, we have to consider the laws of thermodynamics. The first law says that in a closed system, energy can be neither created nor destroyed, only changed in form, which, if you extrapolate it, the amount of energy present at the beginning of the universe will be the same at the end of the universe, just that the form of energy will likely be different. How will the universe end? I put this in just to put something for you to think about. There are a number of hypotheses about what's going to happen to the universe. 3 of them are listed here. One is that the universe will expand forever. We know that right now, at this slice of time, the universe is expanding. Everything in the universe is flying away from everything else in the universe. So one idea is that if this process continues, stars will eventually burn all their fuel, leaving only black holes. So we'll have just nothing but a universe of black holes flying away from each other forever. Or the opposite could happen. The universe could stop expanding and collapse, be a backward Big Bang, in which all the energy in the universe would end up in a tiny little spot. Another hypothesis has to do with dark energy, which is the opposite of gravity in that it causes things to pull apart from each other. So that would be similar to number 1. Anyway, I just wanted to mention these thoughts that people have about how the universe will end, something for you to think about. Second law of thermodynamics. Energy transformation result in increased disorder. It's also called entropy. So that means that every time there is an energy transformation from one form to another, disorder increases. And some of the energy that is released is no longer available to do work. So entropy, the term, is a measure of the amount of disorder or randomness in a system. So the implications of that with respect to life forms are that disorder is always increasing, and that chemical reactions, which is what an organism is, it's just a collection of chemical reactions, they will always go to equilibrium. That means no energy in, no energy out. So when that happens, the organism is dead. So to put another way, to keep an organism alive, there needs to be a constant input of energy. As soon as energy input stops, the organism dies. Now, why do cells need energy to survive? Well, I've pretty much already said that. Cells are subject to becoming disordered due to the second law. They remain alive and healthy only if they have a constant energy input. In other words, we have to eat every day. Electromagnetic radiation. We have to think about this because electromagnetic radiation is the form of energy that is captured to sustain all life forms on the planet. So electromagnetic radiation is a form of kinetic energy. It consists of a spectrum of waves that differ in wavelength and energy content. Waves also have the properties of a particle. Those of you who have taken physics know this. This is always a concept that I have a hard time understanding, how something can be a wave and a particle at the same time. But a form of electromagnetic radiation that is not a wave is called a photon. Photons carry energy, but they have no mass. So you can try to visualize a photon as just a little bundle of energy that's infinitely small. So this little diagram is really important because it tells us what there is about the electromagnetic spectrum that is special for us. So what you see at the top are the wavelengths and the energy levels from low energy on the right to high energy on the left, long wavelengths on the right, short wavelengths on the left. Radio waves are the longest, and followed by microwaves, infrared waves, ultraviolet, X-rays, and gamma rays. Now, all of these different forms of electromagnetic radiation we cannot see, and plants can't see them either. You see this little white gap here in this spectrum? Those are the wavelengths that we can see and that plants themselves can see. So what you see is an expanded part of the spectrum in the lower part, the colored part, and you see a green leaf. So we can see this entire spectrum. Plants can see red wavelengths and they can absorb them. Plants can see blue wavelengths and absorb them. They cannot see green. So instead of absorbing green wavelengths, they reflect them. And that's why plants are green, because they cannot absorb green wavelengths, reflect them instead. So when you look out and you see plants that appear green to your eyes, it's because the plants are reflecting green wavelengths. I thought you'd be interested in what are the functions, how do the non-visible forms of radiation, how do they intersect in our lives? Let's start with gamma rays. They're the smallest, most energetic. They come from various places in the universe, usually very extreme places. They can be generated by nuclear explosions, by lightning, radioactive decay here on Earth. The usefulness for humans is that they are used in cancer therapy. They can sterilize equipment, and they can be used as tracers, doing some types of research. X-rays you know about. You've all had X-rays. They're a penetrating form of radiation. They pass through living things, leaving shadows of bones and organisms. And they are the form of radiation used at airport security scanners, or X-ray machines. UV radiation is found in the sunlight. It converts, it helps produce vitamin D when it hits your skin. It's used for equipment sterilization. I put here coronavirus eradication because you'll often see a claim that a room has been eradicated of coronavirus. And what that usually means is the room has been flooded with UV light for long enough to kill all the virus particles. And we have the visible part of the spectrum, followed by infrared. Infrared we're familiar with from use of garage door openers, TV remotes. Microwaves, we all use them. How do they heat food? By vibrating the fat and water molecules that are in the food. And finally, radio waves, they're the longest in the spectrum from a millimeter, which is very short, to 100 kilometers, which is very long. That would be about 60 miles. So you have to imagine radio waves that are many miles long, and they're used for long-range transmission of radio and TV signals. So how can we capture and make use of the energy that is available in the electromagnetic spectrum? The answer is photosynthesis. I'm not going to say much about photosynthesis in this period, because we'll have a whole section on that later. But just to introduce you to the idea, photosynthesis is a process that's performed by plants that's capable of capturing the energy in part of the spectrum of sunlight. That it's the same part that we can see as visible light. And as we'll get to, the amazing thing about this is that the energy in light, in the wavelengths of light, can be converted into chemical energy. We can't use the energy in light. Plants can. So we get either directly or indirectly all of our energy from plants. Chemical reactions. What is metabolism? It's the sum total of all the reactions in a cell. So at any point in time, cellular metabolism consists of thousands of individual chemical reactions. So because we are alive, we're sustaining thousands of individual chemical reactions, and they're all mediated by enzymes. Each chemical reaction in a living thing is mediated by an enzyme. And the reason for that is that without enzymes, reaction would still occur, but they'd be way too slow to keep life sustained. So we are dependent on enzymes for life itself. So an example of a pathway is here at the bottom. Substances A and B are acted on by enzyme 1. Those are the substrates, converted to a product, C. Then that product can be a substrate for enzyme 2, which converts it to two products, D and E, which then become substrates for enzyme 3, convert them to F. F would then be the end product of this pathway. So there are two types of reactions, endergonic and exergonic. Endergonic ones require a net input of energy, whereas exergonic reactions release energy. And we can visualize that by considering photosynthesis and respiration. A photosynthesis is an endergonic reaction because it requires energy to do its job, to take small molecules and make a large molecule. So photosynthesis takes carbon dioxide and water, makes glucose and oxygen and requires energy in the form of sunlight to do that. A respiration is the opposite. Respiration takes glucose and oxygen, breaks it up into carbon dioxide and water. In the process of doing that, it releases energy. And this energy that's released in respiration is what we live on. That's what keeps us from succumbing to the second law of thermodynamics. So how do enzymes speed up reactions? We have here two illustrations of sucrose, which is a disaccharide composed of glucose and fructose. Sucrose being converted to glucose and fructose. So without an enzyme, it's like pushing a boulder up a hill, a high hill. It takes a lot of energy to do that. But once it's over the hill, a net energy is released from splitting of sucrose. And so it takes a lot of energy. The activation energy without an enzyme is very large. Whereas if we introduce the enzyme sucrase, which is specific for sucrose, what it will do is convert sucrose to glucose and fructose, and the activation energy is now very low. But notice that the net energy released is the same, whether it's with an enzyme or without. So the only difference is that the activation energy is much lower if an enzyme is involved. So what is activation energy's definition? It's the minimum energy needed for a chemical reaction to occur. So I boxed that as something for you to remember.
So excuse me, Professor, if we don't have enzyme, so we need more energy.
Yes, you need a huge amount of energy.
Yeah.
And where would we get it?
Yeah, so kinetic energy, when it's go down, it will be higher.
Right, and chemical reactions are so slow without enzymes that it would be impossible for life to exist.
Yeah, so it's only the difference in energy.
Right.
Yeah.
Yeah, the effect of the enzyme is to reduce the amount of energy that's needed.
Thank you.
So enzyme reactions. This again is an illustration of the effect of the enzyme on the activation energy. And here we're talking about the synthesis of a high energy molecule, glucose 6-phosphate. So the starting substances, the substrates, are glucose and phosphate. And without activation energy, you need a lot, well, without an enzyme, you need a lot of energy to make this molecule. With an enzyme, you need less. So what is the catalytic cycle of an enzyme? So you need to think carefully about this. So we have the enzyme here as a big blue blob, and the substrates as smaller blobs. But what I want you to do is instead of just leaving this enzyme as a blob, imagine what it really is. It's really a tertiary structure of a protein, which we talked about before. And you remember, a tertiary structure is a polypeptide, which is highly folded and cross-linked. And the bonds involved, I've listed here. Remember all the different types of bonds that hold the protein in this configuration. So instead of thinking of it as a blob, think of it as a three-dimensional, highly complex structure. Well, in that complex structure, there is an active site. That active site is where the substrates go. And if they go there and they fit, then a product is produced. But remember, the enzyme doesn't change. So the same structure that you see at the beginning is the structure that you see at the end after the reaction has occurred. Shape is critical for enzyme reactions. And an analogy, a way to think about shapes is to think about locks and keys. So if you look at the lower left here, if you think of a substrate as a key and the enzyme as a lock, if the key fits the lock, the reaction will occur. And on the right of that, you see that the substrate is shaped in the same way that the active site and the enzyme is shaped. So they fit together and a reaction occurs. And on the right, it shows the same thing, except it shows that what happens if the substrate is incorrect. So the correct substrate has two projections on it. If one of them is missing, there's no reaction. So what is the mechanism by which activation energy is lowered? And the short answer is that the bonds in the substrate, the chemical bonds in the substrate, are either broken or weakened by the enzyme. So that's illustrated here in two different ways. If you look at the left, the substrate in green, the enzyme in yellow, the active site, number 1, substrate binds to the enzyme active site. Then 2 and 3, the enzyme itself changes shape so that it stresses the substrate, either weakens the bonds that hold it together or breaks them, so that step 4, it comes apart. The products are released with little expenditure of energy. At the bottom here, you see the same thing. 1 is the substrate and the active site. 2 and 3, the enzyme, in this case, looks like it squeezes it and breaks bonds. So I've listed here the various bonds in protein structure, and among them is a covalent bond. So in what part of the protein structure do we see covalent bonds? Anyone remember? You remember how a polypeptide is made? What kind of bonds hold a polypeptide together?
Peptide bonds?
Do you remember peptide bonds?
Yeah.
Okay, so you have to have a polypeptide before you can have any of the other kinds of bonds. So covalent bonds are important. There's another type of covalent bond involved in tertiary structure. Does anyone remember what that is from this list?
Disulfide bridges?
Disulfide bridges are covalent. Okay.
The hydrophobic attractions.
Say that again, hydrophobic attractions.
Yeah.
Yeah, they are not covalent. Hydrophobic attractions are very weak bonds, very important, but they're weak. They're more like hydrogen bonds. Ionic bonds are stronger than hydrogen, but not as strong as covalent. Okay, the catalytic cycle. Again, we have the example of sucrose and the enzyme sucrase. Sucrose is this blue glob. The active site is shown here, and its shape is critical. And its shape is exactly the shape of sucrose. So sucrose fits into that active site. The enzyme changes it so that it comes apart into its substituents, glucose and fructose, and then the enzyme goes back to its original form. Substrate rate effects. So on this plot, we see on the y-axis the reaction rate. So what is the reaction rate?
The rate at which a reaction proceeds.
Yeah, it's the rate at which substrate is converted to product. So keep that in mind whenever you see the term reaction rate. It's how fast substrates are being converted to products. And on the x-axis is the concentration of substrate. So the reaction rate without an enzyme, it does proceed, but very slowly. And as the concentration of substrate increases, the reaction rate increases, but not very fast. With an enzyme, it increases very rapidly and reaches its maximum rate fairly quickly. So the rate of conversion of substrate to product is going as fast as it can very quickly after the reaction starts. And we have to think about denaturation, because that is one way of inactivating an enzyme. So enzymes can be damaged permanently or temporarily, either way, by exposing them to extremes of temperature, pH, or salt. This is called denaturation. So what happens in denaturation is that the tertiary structure is destroyed. And that's illustrated on the lower right here, where in the picture, you see the tertiary structure of a normal protein. And in the presence of agents such as pH, temperature, ionic strength means salt concentration, or solubility, all the bonds will be broken in this tertiary structure. And so we end up with nothing but the primary structure. So this is just the polypeptide chain held together by peptide bonds. And it has no activity. That's the important thing to remember here, that a denatured protein is an inactive protein. In some cases, a denatured protein can go back together. If you remove the agent that denatured it, it can reconstitute itself back to the tertiary structure and be active again. But I think those kinds of enzymes are relatively rare. Usually if you denature an enzyme, it's done. How can the rate of an enzyme reaction be changed? Well, primarily the three main factors that can change the rate of reaction are the temperature, the pH, and the salt concentration. That would be the ionic strength. Now, what you have here in the diagram are examples of the effect of temperature and the effect of pH change. So let's look at temperature first, at the top. We have, again, the reaction rate, conversion of substrate to product rate on the y-axis, and temperature increasing on the x-axis. So if we look at the typical enzyme from a human, the human body temperature is 37 degrees centigrade. The typical human enzyme has a pH optimum of 37 degrees. If you go above 37, what happens?
Activity drops off.
Why?
The enzyme is becoming denatured.
Right. Remember, temperature is one of the agents. pH, temperature, ionic strength, and solubility. Temperature is one of the agents that will denature. And that's what happens here. The black line goes to 0 as the enzyme falls apart. Now, not all enzymes are attuned to the human body. Some bacteria are heat tolerant. Some bacteria live in hot springs at the boiling temperature of water. They have enzymes that function at high temperature. And an example is what an enzyme called Taq polymerase, which is found in one of these thermophilic bacteria. It can tolerate temperatures as high as 95 degrees centigrade. That's getting very close to the boiling point of water. The optimum for Taq polymerase is 75 degrees. That's where the rate of reaction is the fastest, is at 75 degrees. And later we'll talk about the polymerase chain reaction, PCR, which is entirely dependent on the use of this enzyme, which can tolerate very high temperatures and function at a relatively high temperature. And again, even for Taq polymerase, if you get above 95 degrees, it will denature. On the bottom graph shows the effect of a change in pH. Again, pH going from 0 to 10. Our stomachs have a very low pH, 2. There is an enzyme called pepsin. Anybody know what pepsin is?
I would imagine a digestive enzyme.
It's a digestive enzyme. What would you guess that it digests?
Pepsin bonds.
Does it digest proteins?
Protein.
Yeah, proteins.
Right. It's a peptidase. And it has a pH optimum of 2, which means that it's even active to some extent at pH 1, very acid, and continues to be active up to about pH 3. So that's in a part of our body that has a really extreme environment. More normal would be the pH optimum for trypsin. Now, trypsin is also a protease that's found in the lower part of the digestive system, the pancreas and the duodenum. You may remember this when we talked about the effects of cystic fibrosis. The pH optimum for trypsin is about 8. And the pH of the lower of the pancreas and the duodenum is between 6 and 7. So trypsin is adapted for functioning in a part of the body that has almost a neutral pH.
Does it also break down proteins?
Yes, trypsin is a protease. It does, that's a good point. It does essentially the same thing as pepsin does, but it does it at a completely different pH in a different part of the body.
Professor, I have a quick question.
Uh-huh.
So when an enzyme is denatured, if it's like towards one extreme level, like say for the trypsin, if it was brought into an area of like 1 pH, would it completely just like would it just disappear, or does it stay like till it's moved to a pH level where it function?
You're asking, is it reversible?
Yeah, in a sense, like is there severity to the damage that can be done, like where it's
Yes. So there are degrees of denaturation. So if a protein is slightly denatured, an enzyme, it might be able to reconstitute if the offending agent were removed. But if it's completely denatured, so it's all the bonds are broken except for the peptide bonds, then it's very unlikely that it could recover from that. So if you took trypsin and put it in your stomach, it would probably just get digested by pepsin.
no, okay.
So if pepsin's a protease, why is it pepsin instead of it seems like it would be a pepsidase, not protease.
It is a peptidase.
Okay. Maybe I'm just misunderstood then. Never mind.
Okay, anything else? We can talk about what changes the rate of reaction. And really the reason the reaction rate changes is that we're talking about to what extent can an enzyme be denatured. Enzymes usually don't act alone. They have partners. And you are familiar with some of these. The partners are called either cofactors or coenzymes or prosthetic groups. And you should remember this. The cofactors are always non-protein. So enzymes are proteins, the cofactors are not. So cofactors are actually inorganic, often metals, such as iron or copper or zinc, which are essential for the activity of certain enzymes. Iron, for example, is essential for hemoglobin, carrying oxygen in blood. Coenzymes are not inorganic, they're organic substances, they're not proteins, and often vitamins. So for example, biotin is a vitamin that many people take every day. Some people do. That's a coenzyme. Prosthetic groups are also non-protein. They're part of an enzyme. Examples are heme, flavin, and retinal found in eyes. So when you get a bottle of vitamins from the pharmacy, you'll see a lot of these different things listed. We'll talk about ATP. What is it and how does it carry energy? ATP stands for adenosine triphosphate. It's based on the molecule adenine, which is involved in energy metabolism. But if you remember, adenine is also the A base found in DNA. So this substance has multiple roles. One is in recording our genetic history, and the other is in energy metabolism. So the picture here shows the various different constructions. Adenosine is an adenine molecule coupled to ribose, no phosphate. AMP, adenosine monophosphate, is adenosine with one phosphate group attached. ADP, same thing with two phosphate groups. And ATP has three. And it's the bond in this last phosphate group, the bond between the second phosphate and the third one, that is very high energy. So it takes a lot of energy to make it, and a lot of energy is released when it's broken. But it is the main molecule carrying energy in our cells. So ATP links endergonic and exergonic reactions. And this diagram just illustrates that. You have ADP here at the bottom. Energy in makes ATP with this high energy bond. When that bond breaks, energy comes out and you're back to ADP, but energy is released for cellular work. So this conversion back and forth between ADP and ATP is going on constantly in our cells. And what's important about it is that when ATP is converted to ADP, energy is released.
So does that mean we have a 3 level of energy?
We have what?
3 level of energy. So ATP it's the highest one, and ADP it's the middle, and AMP the first one.
Yeah, AMP is not
the
an energy
lowest.
molecule, as far as I know. And neither is ADP. It's only ATP that has this high energy bond at the third phosphate.
Yeah, so that's what I'm saying. We have 3 level. It's AMP the lowest, and ADP the in the middle, and the ATP is the highest.
Yeah, that's correct. Just don't imagine that AMP and ADP are energy molecules. We don't derive energy from them. We do derive it from ATP.
Oh, thank you.
Metabolic pathways can be divided into two types: anabolic and catabolic. So anabolic ones, think of things getting built up. Anabolic pathways link together simple molecules to form more complex molecules. Anabolic reactions store energy in the chemical bonds of the large polymers that are built up. And these are endergonic, that is, it takes energy to make these large molecules. Catabolic pathways, catabolic, think tear down, so they're the opposite. After a complex molecule is made, a polymer, catabolic pathways break down these polymers into simple forms, and in the process, they release energy. So when bonds are broken, energy is released. And to help you visualize that, I have a couple of pictures here. So anabolic, build up, take monomers, say, for example, glucose molecules, individual glucose molecules, plus energy makes a polymer, which could be glycogen, could be cellulose, but a polymer of glucose It requires energy to make it, but then there's a lot of energy then stored in the bonds that are holding the glucose molecules together. A catabolic pathway, large molecules are broken down into small ones. So if we take a large molecule like cellulose, and it has a lot of energy stored in these bonds, you break them, monomers are released along with energy. So this is how we get energy from food. Right? Most of the food we eat is in the form of polymers. We break it down into monomers, and when we do that, energy is released that our cells live on. Now later, when we talk about respiration, we'll see this diagram again. The food we eat is largely proteins, polysaccharides, and lipids, all polymers. And to make a long story short, all of these polymers get broken down to monomers that go into what's called a citric acid cycle, and there we generate ATP. Well, these catabolic pathways are by and large reversible, and that's shown here on the right. The TCA cycle can go in reverse so that you end up with glucose and glycogen. So this would be an example of anabolic pathways. Going from food to energy is catabolic pathways. And this brings us back to photosynthesis and respiration. And again, we'll go into both these processes in much more detail later, but just to illustrate, to use them to illustrate anabolic and catabolic, photosynthesis is anabolic. That is, it builds up molecules. It makes glucose from carbon dioxide and water, along with an energy input, which comes from sunlight. So photosynthesis is anabolic. Respiration is catabolic because it takes large molecules and eventually makes them into glucose, plus oxygen, makes carbon dioxide and water, and energy is released. Now, I'm not quite finished. I'm going to have to finish this next time because we're out of time, because you have a quiz today. So today's period is 60 minutes instead of 75. And I'll let you go so that you'll have time for to take your quiz. But any questions about anything that we've talked about today? This lecture is not yet in Canvas, but I'll put it there sometime today so that you can refer to it. And also I am recording this.
I have a quick question about denatured proteins.
Yeah.
What exactly happens to them? Like, do they just kind of float around the cell until they're potentially renatured, or is there like a cycle to them?
Well, I'm going to take a guess at what the answer is. My guess is that they're broken down into amino acids, and those amino acids are then used for anabolic pathway, constructing new proteins. Scott or anybody else have any thought about that?
I would imagine, yeah, they're either ubiquinated or they are peptidases come after them and break them back down into amino acids. Because peptidases, I think, typically attack terminal carbons and or break down peptide bonds at terminal aminos, and I think it breaks it down like bit by bit.
Gotcha. Okay, so it's kind of like reused.
Yes, things will be reused inside of a cell whenever possible.
Gotcha. Okay, thank you.
Okay, anything else? I'll finish off talking about oxidation and reduction and the transfer of electrons and how you get energy out of electrons. We'll talk about that next time. If there's nothing else,
Sorry, just a quick question. This isn't on the quiz today, correct?
no, the
Okay.
quiz today is only cell division and what else? Oh, Mendelian inheritance.
Okay, thank you.
And
I
we-
see in Canvas that we have slides for natural selection. Are we going to be covering that next week? Or
No,
are we
not
dropping that?
next week. We're going to finish off this this part of the course that I outlined at the beginning. Oh, you can't see my screen anymore, but the relationship between energy, enzymes, photosynthesis, and respiration, we're going to go through that in detail before we go on to other topics.
Gotcha. Okay, thank you.
Okay. And I'm going to stop sharing. And if there's nothing else, I'll end the session. And everybody do good on the quiz.
Thank you, Professor.
Have a
All
good
right.
day.
See you Monday.